Good evening. My name is Brooke Castle, and I'm also a graduate student at the University of Washington. I study wildfires, and specifically, I study what happens to forests when they have fire, and also what happens to forests when we put the fires out. So if you're interested in hearing more about that, you can come see my talk on April 22nd. So as a graduate student, we receive very specialized training, and sometimes that can be difficult for us to communicate to an audience made up of the public. So the Engage program that we're all involved with has given us some really specific training in, it's training that graduate students usually don't get, which is how to talk about science in a way that's engaging, enlightening, and informative. And you can learn more about this program at engage-science.com. So check out the website. So now to our second guest. Jessie Macadangdang is a graduate student in bioengineering at the University of Washington. Before heading to the West Coast, Jessie received his bachelor's degree in biomedical engineering from the University of Virginia. Beyond science, Jessie is an avid NBA fan, and he also is into sports, although not underwater sports. He prefers sports that can be played under the summer sun. Tonight, he will tell you how scientists can build a heart in a dish to better understand heart disease. So please give a warm welcome to Jessie.
Thank you for that very kind introduction. So tonight, I will be talking to you about how researchers at the UW, and in particular my research group, are trying to build hearts in the lab in order to better understand heart disease, and in particular, find better ways to treat it. Growing up, I loved seeing How the Grinch Stole Christmas on TV. Seeing it on TV normally meant that I was off from school from winter break, presents were on the way, and in general, just a good time to be had to be a kid. Now, the climax of this movie, the Grinch is in a bit of a pickle. All of the Who's presents are about to fall off the cliff if something or someone doesn't come to the rescue. And in the timeless words of Dr. Seuss, and what happened then? Well, in Whoville they say that the Grinch's small heart grew 3 sizes that day. And then the true meaning of Christmas came through, and the Grinch found the strength of 10 Grinches plus 2. And so many of you may say, well, it's the meaning of Christmas is the true hero of that story. But in my own little world, I like to think that it was the Grinch's heart that was the actual hero of the story. So even if your own heart can't give you the strength of 12 Grinches, I want to remind you that it certainly does a lot for you every second of every day. Okay. So in your lifetime, your heart will beat 3 billion times. That's billion with a B. And if that doesn't seem all that special, let's think about what exactly your heart has to do during every one of those beats. Even at rest, your heart consumes more energy than both your legs do if you're at a full-out sprint. And with each heartbeat, your heart pumps blood through a maze of blood vessels. If you were to line up those blood vessels end to end and put them and wrap them around, they could actually be long enough to wrap around the world 2 times. And that's a lot of real estate to cover for every heartbeat. Unfortunately, because the heart has to do all of this work all the time, when something does go wrong with it, it can be really detrimental to patients. Heart disease today is the leading cause of death in the world. 1 out of every 6 deaths is attributable to heart disease. That's more than cancers, that's more than all infectious diseases combined. Additionally, the impact of heart disease can be felt in every single one of our pockets. In 2010 alone, heart disease cost Americans $444 billion. And that's only going to keep growing as our society continues to battle diabetes and obesity. And so Americans today are living longer with heart disease and also a lower quality of life. And in and of themselves, these socioeconomic impacts of heart disease certainly merit continued research and investment in studying, how can we help patients who have heart disease better. But for me, and for, I'm guessing, many of you in the room today, heart disease can be much more personal because 1 out of every 6 of these people could be someone that you care deeply for. When I was 12, my grandfather had quadruple bypass surgery, and just this past year, my grandmother suffered a small heart attack of her own. So with so much riding on trying to find better ways to treat heart disease, it's important to understand the limitations that currently face us when we study heart disease and trying to find new treatments for patients. Traditionally, and probably most obviously, you study heart disease in patients. Someone will come into the clinic presenting with symptoms. Their doctor will look at them and do certain tests. And then that patient could be asked to join a new clinical trial to see if they want to try out a new drug for their particular disease. But there are pretty tricky ethical considerations when you have to do research in patients. For one, no one wants to feel like a guinea pig in a lab. Everyone wants the best treatment possible for their own loved ones, and in particular themselves. And so no one wants to be getting that untested drug. They want the best possible treatment for themselves. And 2, clinical trials are just really expensive. It costs billions of dollars to take a new drug from the pipeline into the clinic or into the market. And it really is the last place that drug companies want their drugs to fail. And so as an alternative approach to studying heart disease in patients, you can also take some of the ethical considerations out of the equation by studying hearts isolated from patients or small pieces of heart tissue. And so these hearts could come from heart donors, or they could be grown in the lab. And so donor hearts, unfortunately, are in extremely short supply. Every year there are about 20 million patients that have heart failure. And these are the patients that are waiting in line for these transplant hearts in order to save their lives. But only about 3,500 donor hearts become available in the entire world every year. So the vast majority of these patients are waiting on these transplant lists for these donor hearts that probably are never going to come. Additionally, as researchers, we want to be able to poke and prod and do all types of crazy experiments on these hearts and on these heart tissue to see if we can find that new miracle drug or that new miracle treatment that will help all of these patients. So the question kind of remains, if doing research in patients is expensive and unethical in certain senses, where are we going to get these hearts in order to do this type of research on them? And so one thing I want to remind everyone of is that at its simplest level, the heart is a muscle. And so as such, it's made up of muscle cells, about 3 billion to be in fact. And so these muscle cells are like the small building blocks that make up the heart itself, and cells actually make up all the organs in your body. And like many things in science, if we can break things down into their simplest components, then we can start answering some of the unknown questions at hand. So instead of having to wait for donor hearts to become available, what if we could just build our own hearts out of these small building blocks to do the types of research that we want to do? But you may be saying to yourself, well, where are we going to get these heart cells if donor hearts are already in short supply? Because I know I'm not lining up to have someone take out a portion of my heart to put in a dish to do research on. And this is where stem cell technology really comes to the rescue. And as Christina said earlier, stem cells are really specialized in that they can turn into any cell type that we want them to. And so if we give them the right cues, or in the right words, as Christina said, we can turn these stem cells into the heart cells that we're really interested in looking at. And that these cells act act very similar to the way your own heart works. So your heart works to pump blood to your whole body. And so these stem cell-derived heart cells actually beat the same way that your own heart would. And so that their job is to contract. So when we grow these stem cell-derived heart cells in the lab, this is what we look like. This is what they look like. They're much smaller than anything you can see with the naked eye, so we have to use microscopes in order to look at them. And if we use normal light, like the light you would get out of a flashlight, this is what they look like in a dish. And if we use specialized lasers and specialized equipment, we can actually make certain aspects of these cells glow so that we can understand their function a little bit better. And in particular, in this picture here, blue would be the nuclei of these cells, and that's where all the genetic information is stored in these cells. Now you may be saying, well, okay, if we can make heart muscle in a dish, now how close are we to actually curing heart disease? And so one thing that I haven't quite mentioned yet, and that is a major obstacle in using stem cell-derived heart cells, is that they are what we consider immature. So as an adult heart cell, the heart cells in your own body, they're big, hulking bricks of muscle. Their job is to contract every second of every day to pump blood to your body. Stem cell-derived heart cells, on the other hand, are not so much. They're pretty wimpy in the fact that they can't beat very hard, and they don't look anything like an adult heart cell would look like. And this is important when considering if we want to study adult heart diseases, we really want to be looking at adult heart cells. And so this maturity issue is really not all that surprising when you think about it. It takes your own heart years and years and years to develop into the organ that we all have in our chest right now. When we turn stem cells into heart cells, we do it in probably a little less than a month. And so it's not all surprising that these cells are not quite as mature as an adult heart cell are. So a lot of research is spent, a lot of time is spent trying to figure out how can we turn these immature stem cell-derived heart cells into the adult heart cells that we're interested in. And so people try all types of approaches in order to speed up this clock. They'll shock these cells with electrical signals. They'll culture them with other cell types to see if they'll act like mentors to mature them a little bit. They'll pull on them actually to see whether or not they can kind of get them to act appropriately and turn into what we want, which is an adult heart cell. Now my lab takes a little bit different of an approach. Instead of trying to figure out how can we shock these cells into becoming more mature, we try to think about how are we growing them. And we actually try to trick the cells into thinking, hey, I'm growing inside an actual heart, and I'm not growing inside of a dish somewhere. And we do this because, like anything else, when you place something in a foreign environment, it's never going to act the way you want it to. For instance, I don't think this buffalo is going to be sitting down in those chairs anytime soon, nicely. I'm pretty sure it's probably going to rampage in this room pretty soon. And this is obviously not its natural habitat. The same thing goes for heart cells. When we grow heart cells traditionally in the lab, we grow them in petri dishes. And this is just how cell culture has always been done. We always grow cells inside these petri dishes and give them medium. But to a stem cell-derived heart cell, these petri dishes kind of look like this flat, barren wasteland that they haven't ever experienced before. There's nothing really for them to interact with. Because stem cell-derived heart cells are really used to a kind of more complex environment. And so they're used to having lots of things to grow on and lots of things to interact with, kind of like a jungle. So this environment, which we call the extracellular matrix, is kind of like a jungle gym that these stem cell-derived heart cells, or these heart cells in general, can interact with and can grow on. And what we do in my lab is we try to mimic and recreate the cell's natural environment. And we want to be able to give the cells something to interact with, something that they can grow on. And so we do this by building these nano-ridge devices. And these nano-ridge devices are pretty simple conceptually. They're basically these rows of ridges that are kind of like railroad ties that the cells can sit on top of and they can sense. And now these nano-ridges are small in the fact that you can't see them with the naked eye per se, but you can definitely notice that they're there. So this surface is a nano-ridge device, and it has this cool iridescent appearance. And this iridescent appearance is the same reason that is there for the same reason that you see the same type of thing on the backside of a CD. It's just the light interacting with these small features on top of this device that cause it to kind of diffract and have this cool rainbow pattern. And so our hope is that by putting our stem cell-derived heart cells on top of these nanoridges, that they'll be happier and that maybe they'll act the way we want them to and turn into cells that are more like adult heart cells. And the results we get are pretty cool. When we grow stem cell-derived heart cells in a petri dish-like environment, they basically become randomly oriented. They're kind of all over the place, not doing what we really want them to, and they're kind of disorganized. But when we grow them on our nano devices, the cells kind of pop into alignment, which is a really good thing. And so they start to look a little bit more like the adult heart cells that we're really striving for, which is a good thing. And so just by putting these stem cell-derived heart cells in a more familiar environment, we can actually boost their maturity and turn them from a more immature state into a more mature state. So this is what we're left with when we do my research. Basically, we get a small piece of heart tissue in the lab that will beat and contract in a single direction. They're pretty much lined up. And it's like a small piece of a mature heart tissue. And so through the microscope, we can see the cells beating in unison in a single direction, which is a good thing. And so this piece of tissue is more mature than other types of stem cell-derived heart tissues, and that can be used for a lot of important research and treatments. For example, as Christina mentioned before, these small pieces of heart tissue could be used kind of like a biological Band-Aid. So when you have a heart attack, a small or a portion of your heart is damaged, and that's never going to regrow because we don't have heart stem cells per se. But if we take our beating piece of heart tissue and put it on top of that damaged area, it's possible that we could regrow that heart so that that person can have a better quality of life. Alternatively, what we can also do is start doing some really intricate and better drug screening. So instead of having to wait for heart donors or enroll patients in a clinical trial, what we can start doing is giving these small pieces of heart tissue that act like an adult heart and see how these new drugs affect them, whether or not they hurt them, whether or not it's good for them. And so we can do really intricate testing, which we couldn't do before. And this kind of overcomes the obstacle of not having enough donor hearts to go around. And so because we have a better heart muscle in the dish, we can find safer and more effective therapies. And with that, I'd like to thank everyone that made this talk possible, including the ENGAGE program and all my collaborators at UW. And I'll open the floor for questions.
Short question. Your visuals show the beating heart in a dish. Does that exist? How close?
So it's definitely not a full heart. We're not making the full heart that maybe you saw on the opening slide. What we are growing are these small pieces of heart tissue. which are very real. You know, I look at them every day and I work with them every day. And so the hope is that before we can, you know, regrow the entire organ, let's grow small pieces of it that can eventually maybe be used to help patients that have heart disease.
So basically, you're talking about using these cells as models, test models. Have you been able to validate it in any way, throw any chemicals in there, some DIDs or something, and see if they actually respond to the...
Yeah, we do that all the time. So there are definitely known drugs that you can give these cells that have a certain, you know, negative effect in a person's heart. And so we can kind of correlate that with that small piece of heart tissue's function. And we definitely see the types of, you know, the types of response that we're expecting. So this is actually, you know, we're making small pieces of heart tissue that are looking and acting like, you know, a heart that, you know, everyone else has. So it's something that is definitely, you know, next to be used.
So you were talking about plating the cells on these nanoridges. So if you were going to put them in the patient, would the nanoridges be in the patient as well? And if they are, do you think that would be harmful? Or, and if they're not, if you take them off the nanoridges, do you think they're going to still maintain that same, I guess, stacked structure? Or
Yes. So our lab is using what's called cell sheet engineering. So we can grow our cells on top of these nanoridges, have all of the positive influence that they you know, would exhibit on these cells, and we could basically pop the cells off, and that they'll maintain their alignment. They'll maintain what we would consider a more mature phenotype, or, you know, they look like an adult heart cell. And so then that could be implanted on top of, you know, in a damaged heart. Or alternatively, we can actually make these nanoridge devices out of all sorts of materials, including biodegradable materials. So it's possible we could make, you know, like a patch or a Band-Aid that has cells on top of it and that are basically have these nanoridges, and then we put on top of a patient's heart to help regrow it. So both.
Do these stem cells, when you put it on the heart, do they just keep reproducing at some point, or is something in our bodies tell them to stop, or what goes on in that?
So these stem cell-derived heart cells, they have a little bit of regenerative or proliferative capacity. They can keep growing, but they actually are much reduced compared to a stem cell like Christina was talking about. So there's not that much worry that they'll, you know, grow too much, and that maybe we do want them to grow a little bit and maybe, you know, add more heart cells to there. But there's no real signals that we know of right now that if we just put them into the heart that it'll automatically shut off or work exactly right. So the hope is that, you know, with Christina's research, maybe we can understand, you know, how these stem cell derived, you know, tissues and muscles are working so that we can better control them.
How big have you guys been able to grow the heart cells? They're kind of like the Band-Aid. How big is the Band-Aid?
So we can actually make it what we would consider macroscopic scale, so a couple inches wide. So as big as we can make our nanoridge devices, that's how big the Band-Aid which we can make. And so we have the ability to make it, you know, something that would basically cover the entire surface of the heart if necessary, or it could be smaller. So it's really dependent on how big our nanoridge devices are, which we can make pretty large.
So have you actually tried taking those cells and putting them in a real heart? And do they automatically pick up the beat of the other heart cells around them? Or do they continue in their own rhythm?
That's a very good question. Whenever you put anything that is outside of the body into your own body, you have to make sure that it's compatible. And so we have placed these patches on rat hearts. hearts. We're not going into patients yet. And it's promising that these human stem cell-derived heart cells can actually keep up with a rat heart, because a rat's heart is just going to beat way faster than our hearts or whatever. So we have been able to place it on top, and it hasn't caused arrhythmias or misbeats in these rats yet. So it's something that is promising.
And do they continue to live? They don't like die off early or something because,
Yeah, and so there are, this is early on. What we found is that they remain there for as long as we run our experiments. We can definitely sense that they're there. How long they'll be, whether or not they're actually engrafting or growing into the new heart, is kind of remains to be seen. But it's something that is, of current research, and we think that we're actually getting, that muscle to regrow, so that it's helping a patient that would have some sort of heart attack.
And is there a limit to how thick you can make it?
Yep, they're definitely, so you have to consider how is this tissue or this muscle going to be supplied with oxygen. And so we can make it, we could theoretically make it as thick as we want, but if we made it that thick, then it wouldn't survive. So if we make it, a couple microns thick, which would be a couple layers of cells, then that'll survive, and it'll be able to have blood supply. As far as the actual thickness that we can, maximum thickness, I'm not entirely sure.
Thank you.
Yeah.